it cleaves uh, the DNA into fragments at either near specific recognition sites with the molecule known as a restriction site. Means this is the endonucleases. They is cut into specific sites it will cut, and that endonuclease we call as a restriction endonuclease. Restriction sites we choose a specific site or recognized sites which is used to cut into small pieces. They have the capacity to recognize specific base sequence on DNA and then to cut into each strand at the given places. Okay, so the DNA sequence cut into small pieces that recognize the sites. That is the DNA double helical structure. We know that one. Hence, they are called as the molecular scissors. Why we call as the scissors here? This is the cut into small pieces. These restriction endonucleases we call as scissors. Why we are called as scissors means the DNA double helix strands with help of the restriction enzymes or nucleases cut into small pieces. So it will act as a scissor-like function. So this is the see here that picture. That is the codon is the DNA codon is there. Huh? That is the means pyrimidine cytosines are there. So this one is cut into some that is the restriction enzymes means gone into adenine, cut into small pieces. Okay, this one is very helpful in the molecular biology. In when we need the specific sites, it is very useful to the identify the sites and cut into small DNA fragments which are involved in the transmission of the transmission to the characters to others. Source of restriction enzymes. What are the source? The natural source of restriction endonuclease are we know that one bacterial cells only. So that is the bacteria. They have the plasmids are there. These plasmids also they are some vector activities. So mainly the endonuclease restriction endonuclease obtained from the bacterial cells only. These enzymes are called as restriction enzymes because these restrict infection of bacteria by certain viruses, example bacteriophages, by degrading the viral DNA without affecting the bacterial DNA. So thus this function in the bacterial cell uh, is to destroy the foreign DNA that might enter into the cell. These restriction enzymes recognize the foreign DNA and cuts it at several sites along the molecule. So that's why the DNA enzymes, which is recognize the foreign element that cut into small pieces means we have to choose the different sites, different areas. So that one is to cut into small molecule. Each bacterium has its own unique restriction enzymes, and each enzyme recognizes only one type of sequence. So each bacterium has its own unique that is the restriction enzymes, and each enzyme which is recognized only one type of sequence is present. So recognition sites. How we are to recognize these sites? The DNA sequence recognized by the restriction enzymes called as the palindrome, palindromes. So these palindromes are the base sequence that is read the same on the two strands, but in opposite directions only. So this DNA sequence recognized the some restriction enzymes that one we call as a palindromes. So for example, if the sequence on one strand that is the G means guanine, adenine, adenine, thymine, thymine, cytosine means it is read into 5 to 3 direction. The sequence on the opposite strand here, cytosine, thymine, thymine, adenine, adenine, guanine, read into that is the opposite direction, 3 to 5 direction. But where the both strands are read in the 5-3 direction, the sequence in the same. So here that is the palindromes appears accordingly. What here 5 to 3 direction here guanine, adenine, adenine, thymine, thymine, and cytosine. And 3 to 5 direction here opposite direction is appeared. In the addition, there is a point of symmetry with the palindromes. In the example, this point is in the center between the 80 means adenine thymine, adenine thymine. Okay. The value of restriction enzyme is they make the cut in the DNA molecule around this point of symmetry. Some enzymes cut straight across the molecule at the symmetrical axis, producing the blank. This is the producing the blunt ends. So of more value, however, the restriction enzymes that cut into between the same two bases away from the point of symmetry on two strands, thus producing the uh, staggering break. So next one, wait. అందుకో ఏం సౌండ్ డోర్ పెట్టక మామూలుగా పడుకో Uh, the mechanism of the cleavage of restriction enzymes. Yeah, when a restriction endonuclease recognition a particular sequences, we choose that particular sequences, that is adenine R3 to 5 direction or 5 to 3 direction in spins through the DNA molecule by catalyzing the hydrolysis. So hydrolysis, we know that one. That is the splitting of mechanical, means a chemical body, that chemical bond by addition of water molecule. So of the bond between the adjacent nucleotides, to cut the DNA, all the restriction enzymes make into two incisions, once through each sugar phosphate backbone, means that each of the DNA double helix. So some of the types of restriction enzymes are there. So types of restriction enzymes, see here in the traditionally four types of restriction enzymes are recognized, which is designated as 1, 2, 3, and 4. Means type 1, type 2, type 3, and type 4, which is differ primarily in the structure, cleavage site, specificity, and cofactors also. So this one is the that is the primary means structure of the restriction enzymes, cleavage sites, and their characters and cofactor. What are involved in the cofactor? These are based on these characters. Here we are to categorize it into four types of the restriction enzymes are present. Okay. So what are there here? Type one. In the type 1 enzymes means type 1 restriction endonuclease, which cleaves at the sites remote from the recognition site. They require both ATP and S-aldolase L-methanamine to function. That is the multifunctional protein with both restriction and methylase activities. This one. And type 2, there is a cleavage within the DNA or at the short specific distance from the recognition site, or either most require magnesium, either single function enzymes independent of methylase. The type 3 enzymes. What are the type 3 enzymes? These cleavage at the site, a short distance from recognition site, require ATP but do not hydrolyze it, and S adenosyl L-methanamine, methionine L-methionine stimulates the restriction but is not required. It exists as part of complex with the modification methylase. These are there. And another type 4 enzyme, this is the target modified DNA. Here, example of methylated and hydroxymethylated and glucosyl hydroxymethylated in that methylated DNA. So nomenclature of the restriction enzymes. How we are to give their names of the different functioning restriction enzymes. With the help of their names only, we are to give their names. So, and in 1970s, the many restriction enzymes has been identified, while the type 2 restriction enzymes, which is characterized in 1970. So each enzyme is named after the bacterium from which it was isolated, using the naming system based on the bacterial genus, species, or strain. For example, name of the E. coli restriction enzyme. That one is obtained from the E. coli. Uh, RI means restriction enzymes are restriction endonuclease. So that is the E. coli. E means Escherichia, and CO means coli specific. That is the specific species. R means RY113 strain. I means the first identified or order of identification in the bacterium. So another one is the applications. Applications of restriction endonuclease. That is the restriction enzymes can be isolated from the bacteria cells and used in the laboratory to the manipulate the fragments of DNA. Such those that contain the genes for this region, they are the indispensable tool for the recombinant DNA technology. That the genetic engineering, we call that one genetic engineering or molecular biology. This is the restriction endonuclease is more important in these technology or DNA technology or genetic engineering. So most useful aspect of the restriction enzyme is the each. So the most useful aspect of restriction enzymes is that each enzyme recognize the same unique bases, that is the unique base sequence regardless of the source of DNA. It means that these enzymes established fixed landmarks along an otherwise a very regular DNA molecule. This allows dividing a long DNA molecule into fragments that can be separated from each other by the size with the technique of gel electrophoresis. Each fragment thus the generated are also available for further analysis, including the sequencing. And what value of cutting DNA molecule up into discrete the fragment is being able to locate a particular gene on the fragment where it is residues, which is done by the general technique of southern blotting technique. So see here some of the I shown to so the picture some of the examples of the uh, restriction enzymes means restriction endonuclease. So for 5 direction here adenine, adenine, guanine, cytosine, thymine, thymine, and which is to handle the digest. What happen here that will be divided. Okay, clear. The seeking of producing protruding the ends by 2 or 3 duct are blunt ends but produced by a specific restriction enzyme. So some of the here one of the most popular restriction enzymes is called that one is the E. coli obtaining enzyme ECORI. So that is the popular restriction enzyme which is obtained from the E. coli bacteria. Already we have discussed that one. E. coli bacteria. So 100 of these restriction enzymes with a different sequence, that is the specific sites have been isolated from several bacteria and are commonly commercially which is available. So what is that one? EcoRI, EcoRI2 and BAMHI and another one Hind third, that is HIND third. So these are the some restriction enzymes is very useful in the DNA technology. Okay. So, sir. So, now, the next class we have to discuss the uh, modifying enzymes. Modifying enzymes. Here, so I'm trying to wait sometime. So here, so this is the modifying enzymes. Some different types of the modifying enzymes are present. We have to discuss one by one. So, what is the modifying enzymes which are used in the molecular cloning, molecular biology or cloning techniques, DNA? These restriction enzymes and DNA ligases represent the cutting and joining the function in DNA manipulations. So this one is cut just like a scissor like and DNA ligases which is represent cutting or giant function of the DNA molecule. So all these enzymes involved in genetic engineering fall under the broad category of enzymes that one we known as the DNA modifying enzymes. These enzymes are involved in the degradation, synthesis, alternation of nucleic acids. So DNA ligase is the important molecular enzyme as its function is to repair broken phosphodiester bonds that may occur at the random level or as the sequence of DNA replication or recombination. In genetic engineering, it is used to the seal discontinuous in the sugar phosphate chain that is raised when the recombinant DNA is made by joining DNA molecule from different sources. It can therefore be through as the molecular glue, which is used to stick pieces of DNA together. This function is crucial to the success of many experiments, and the DNA ligases is therefore a key enzyme in the genetic engineering. In the genetic engineering, we already discussed that one. This one is the very important in the genetic engineering aspects. Okay. So the DNA ligase is an important cellular enzyme as its function. It is to repair broken phospho. There is the enzyme. So, what are the restriction endonuclease? They are also called as that we know that one. That is the endonuclease. So, types of some DNA modifying enzymes. What are there here? The nuclease enzymes degrade nucleic acid by breaking the phosphodiester bond that the hold the nucleotide together. The restriction enzymes are good example for the endonuclease, which cut the DNA strand. A second group of nuclease, which is degrade DNA from the uh, terminal of the molecule, which is known as the exonuclease. Apart from the restriction enzymes, there are the four useful nucleases that are often used in the genetic engineering. This is called as these are the what BAL31, exonuclease 3, and deoxyribonuclease I, and S1 nuclease. So these enzymes differ in their prices, mode of action, and provided the genetic engineer with a variety of strategies for attacking the DNA. The nuclease BAL31 is the complex enzyme. Its primary activity is the fast acting three-strand exonuclease, which is coupled with the slow acting endonuclease. When the BAL31 is the present at the high concentration, this activity is effectively shorten DNA molecule from both termini. Another is the exonuclease. Third is the exonuclease is that generate a molecule with the protruding the five termini. The DNA is the one cut there either single strand or double strand DNA at essentially random sites. The nucleus S1 is the specific for the single strand DNA, either RNA. Another type is the polymerases. So polymerases is the enzyme is synthesize copies of nucleic acid. This is used in many genetic engineering processes. When they describe a polymerase enzyme, that one the DNA dependent or RNA dependent may be used in the indicate the type of nucleic acid template that the enzymes use. The DNA polymerase 1, the enzyme DNA polymerase 1 has, in addition to its polymerase function, 5 to, uh, 5 to 3 direction and 3 to 5 direction exonuclease activity. These enzymes catalyze and stands replacement reaction where the 5,3 exonuclease function degrade the non-template stand as the polymerase synthesis the new copy. A major use of this enzyme is in the translation procedure for radio-labeled DNA. The 5,3 direction exonuclease function of DNA polymerase 1 can be removed by the cleaving the enzyme to produce what is known the uh, fragment. This retain the polymerase 3 to 5 exonuclease activity. The Trino fragment is used where a single strand DNA molecule need to be copied because the 5 to 3 exonuclease function is missing. The enzyme cannot be degraded the non-template strand of double strand DNA during the synthesis of new DNA. Here, the major use of these fragment means fragment is include radio-labeled by primate synthesis and DNA sequencing by deoxy method in addition to the copying of single-strand DNAs during the production of recombinants. A modified form of DNA polymerase, one is called as the um, fragment. It is useful polymerase that is used in widely number of applications. So another one is the RTase means reverse transcriptase. So reverse transcriptase is also very important modifying DNA. So reverse transcriptase is the RNA dependent DNA polymerase, and therefore the producing a DNA strand from RNA template. It has no associated exonuclease activity. The enzyme is used mainly for the copying mRNA molecule in the preparation of cDNA or complementary DNA. We know that copy DNA also sometime. For cloning although it will also act on the DNA templates. So reverse transcriptase is the key enzyme in the generation of cDNA, complementary DNA. The enzyme is an RNA-dependent DNA polymerase which produces a DNA copy of an mRNA molecule. The enzyme that is modify the ends of DNA molecules. The enzymes alkaline phosphate or polynucleotide kinase and terminal transferase act on the terminal of DNA molecule and provide the important function that are used in variety of different ways. The phosphatase and kinase enzymes, as their name suggested, that are involved in the removal of or either addition of phosphate group. So when the bacterial alkaline phosphate, uh, there is also the similar enzyme. So there is the enzyme is used to prevent the unwanted uh, ligation of DNA molecule, which can be used, can be problem in the certain cloning procedure. It is also used prior to addition of radioactive phosphate to the 5' of DNA by phosphonucleotide kinase. So terminal transferase, which is repeatedly adds a nucleotide to any available three terminates. Although it works best on the producing three ends, conditions can be adjusted to the blunt end or the seized molecule may be utilized. So another one is the vector. Another type is the vector, that is the cloning vector. The DNA molecule is used as a vehicle to transfer the foreign genetic material. So here we are to discuss some of the vectors are the plasmids, bacteriophages, cosmids, and that one on east artificial chromosome. That one we call as the YS, yes, YAX. YAX means east artificial chromosome, bacterial artificial chromosomes, YAX and BAX. So the comparison vector available for the cloning fragmentations. What is here? Different plasmids, phase one plasmids, YAX and BAX. So vectors for yeast and other fungi. The yeast Saccharomyces cerevisiae is one of the most important organisms in the biotechnology or either genetic engineering. The development of cloning vector for yeast was stimulated by the discovery of the plasmids that is present in the most strain of the Saccharomyces cerevisiae. So why it is the popularly used? The yeast cell is much easier to grow and manipulated than the plant and animal cells. The cellular biochemistry and regulations of yeast are very like those of higher eukaryotes. These are many yeast homologue of human genes, those involved in the cell division. So the yeast can be a very good surrogate and host for studying the structure and function of eukaryotes gene producers. So the different kinds of cloning vector is divided from the yeast. That is the yeast episol plasmids, yeast integrated plasmid, yeast is replicative plasmids, yeast artificial chromosome. That is we already, yeah, okay, we know that one.
Mami.